come and help and present this for students' benefit. So please utilize this opportunity very well. At the end of it, please post questions in your chat box or Q&A, and sir will answer it in the given time. Thank you very much, sir. Welcome. Thanks for accepting on behalf of DNB board, student, NBMS, and me and students. We really thank you. Welcome, and over to you, sir.
My pleasure. Thank you so much, Dr. Somsekhar, for your kind introduction. And as I might start saying that, during our curricula, I mean, for the last 50 years that I remember of, and even till today, research, clinical research is never taught in our undergrads, post-graduation, even for super specialization levels. All that we have been doing has been of our own and trying to learn whatever best we can. Now, I think there is an initiative by the government, and I think the National Board of Examination is also looking into it as to how research could be taught right in the beginning, because what we understand so far is, well, we have to go for our thesis during our post-graduation and try to get it as quickly as you can, get any question, just run it away and you are done. So while we will be discussing, we can't discuss whole of research and the clinical trials that are possible, but I'll try to touch base upon the basics and fundamentals that are required to be known, at least be aware of, when we are running our postgraduate or super specialty programs. Now, this may apply to, now since this is an oncology audience today, but what I will be talking about will be more broader and general regarding research as such. And, please post your questions as and when you have an opportunity, and I'll try to address those at the end of the session today. So I will be speaking for about 40 minutes, 45 minutes, and then maybe we'll have about 15 minutes to discuss about our questions. So since this is addressing to the audience of, oncology, so almost every, all of us know that almost every cancer treatment available today has come about because of clinical trials. When we are looking at new treatments, new immunotherapies, we are talking about CAR T therapies, we always refer to the journals that are available, what is the latest in that, and we refer to that, and that becomes our standard. And ultimately, protocols are developed and standard of care is developed based on that. So as we know, every cancer treatment has come out of clinical trials, and these are carefully designed research studies. When we say design of a research study, it's extremely important that how you are doing that research so that it can lead to the conclusions that we are looking at. And so that such as cancer fighting drugs, to see how well they work. Because if the study is designed well, then only you will be able to get the right answers, and that is applicable for our own patients. Now, what are the different types of cancer trials? There are 4 different categories, and these are very general understanding, broad understanding. Even a layperson can understand what it means. So clinical trials for new treatments. So we are trying to look at most clinical trials are done to test new treatments, whether one treatment is better than the other. So it might be for a new drug, a new combination of drugs, because in chemos we try to mix multiple drugs and see what is the best. A new way of doing a surgery or giving radiation therapy, for example, HIPEC treatment, whether that works better or not, that's what we try to do. Or a new way of giving treatments, the methodology of adopting treatments, HIPEC again being another example. And changes such as to nutrition and exercise that can help people live longer. Sometimes we try to see what are the other things that are important when we are treating cancers. Now, nutritional status of cancer patients is extremely critical. And not only cancer patients, but it's important for many other diseases as well. So we try to evaluate how treatments work under these different types of evaluations and studies that we do. So whenever we arrive at this final treatment, we call it as a standard of care. So typically we say we are going to compare the standard of care versus treatment option 1, 2, and 3. And that's how we try to design and try to answer our questions in our day, which will be applicable for our day-to-day cancer practice. Another set of trials is to prevent and look for cancer. Sometimes we try to evaluate and are done to find new ways of prevention cancer, reduce cancer risk. And this is important because treatments often work better. And these kinds of, how can a kind of cancer be prevented, or can the risk of this cancer be lower compared to what we know today. Is this cancer inherited? Are there any genetic predispositions to a particular type of cancer? And is there a way to prevent inherited cancers from developing? And now genomics is doing a big job in this aspect. How can people be aware of this risk? How do you communicate and disseminate this information that we have learned out of our research? And can the risk of cancer be reduced by eating or avoiding certain types of foods, by taking or avoiding certain medicines? Does it help to make life changes such as getting more sleep or exercising? We're talking about quality of life, overall risk reductions and things like that. So these are another set of trials that we try to look at. When we say trials, I would say we're trying to get answers to these questions that we face in our day-to-day practice. Similarly, clinical trials for symptoms and side effects during treatment. We talk about nausea and vomiting of chemotherapy. How can we address that? That's important for us, and we want to find out what are those things which can reduce and make their life better as a result of clinical trials to develop anti-nausea drugs, for example. And similarly, then late effects. Many cancers are cured through treatment now, but cancer treatments can cause side effects many years after you are cured. All the long-term and late effects. Does it have any cardiac toxicities over a long period of treatment when we are giving, today we take cancer treatments are given very early on. It's not like when we are having metastatic diseases and then we give chemo, right from phase 1, we are trying to cure cancers. Now, when we are giving long-term treatments, does it have any impact? Subsequently, we turn to look at, outcomes based on that. So some of those, so these are basically when we talk about type of trials on one hand, but at the same time, we're trying to tell these are the questions that we come across on a day-to-day basis in our oncology practice, and how do we address some of these which can make the lives of our patients better. So this was just a background of what we come across in cancer treatment. And now what I'm going to tell you is about, in order to understand what are we going to cover today. This was very brief I wanted to give you, but what we will try to cover is basically an overview of clinical product development, because that will give you an understanding of how products, whether it's drug, devices, therapies, surgical techniques, how are they developed and how is that process happening? What is the overview of the clinical trial process? What are important aspects, like what Dr. Somsekhar said in the beginning? What are those different types of processes that are required in a clinical trial? And what should we be more concerned about? And then overview of subclinical study designs. We'll talk about very broad, we can't get into the deeper details of those, but just touch base on some of the different types of designs that we typically run in clinical trials. And some of the basic study protocol. I mean, what should a protocol look like, which we call as the methods and materials and methods. Typically, when we look at articles and journals that are published, that are basically based on certain protocols based on which we design trials. So these will be some of the basics that we will cover today, because this will give you an understanding of how the whole process of research, drug development, and the regulatory aspects associated with that, what are the processes that are required for that. Now, to give you a basic of the research and development landscape, I just wanted to, before we start that, two disclaimers is that, well, this is those who have knowledge don't predict, and those who predict don't have knowledge. That's Lao Tzu, who had called this in the 6th century BC, who was a Chinese poet. He mentioned this. But while I'm going to make one disclaimer that, all this information that on the slides that I'm presenting is a mix of multiple presentations that I have done in the past, and some of them are in public domain. All of them are not my personal slides that I have developed. They have been taken from other presentations which are in public domain. Now, when you look at the drug development process, different stages of drug development, if we have to really look at it from a preclinical research, which is what we call as, when we have a product which has been synthesized and purified, and, we call it a lead generation in the whole clinical development process. Now, which is only the preclinical, which is happening in the labs, or whether with, in petri dishes or in the laboratory setting in vitro, and then in the animal testing study before it comes to the clinical studies. And then it comes through a regulatory process of development, which is the regulatory agencies like the FDA, the DCGI, and different regulatory agencies who ensure that basically that this process of whole research and development of a particular product has been done as per the standards that it is expected to be, so that it builds public confidence and assurance of public. And that's where the regulations come into place. So typically, the whole process is basically a preclinical research process, the clinical studies, and then it doesn't stop there. You have a new drug application, and then there is a post-market piece, which we have to really run through our whole clinical development process. So the product life cycle of any product, like if we talk about any product, as an example, like let's talk about gemcitabine, which has been such an old drug, and the chemotherapeutic other drugs, they have been there for like 20, 25, 30, 40 years. And these products continue to be in the market, and these have to be regulated and followed up. And it doesn't end there. So the whole development process is a long process. Now, in terms of time, and sometimes this, why I'm talking about this whole procedure is that the amount of time that it takes to develop one particular product till it actually comes to us for giving it to patients is a long haul. It takes about 10 to 12 years and different time periods when it is evaluated, when we go from phase 1, phase 2, phase 3, different types of clinical trials, and then when it comes to the launch. So it's a long process of developing drugs. So it's not very easy. And when you when you start with the beginning of discovery, you register a patent. And a lot of times we do talk about drugs which are available in the US, why are they so expensive? They are patented drugs because a lot of effort and money goes into these developmental products, and then failure rates that we have, we'll talk about it in the next slide. So it really takes a long haul to develop these particular drugs. So basically, when we talk about life cycle, so it again a replication of what I mentioned earlier on. There's a research which is basic research, applied research, and then we come into the developmental part, which is the preclinical research, and then the clinical trials. And then the patentee comes and commercializes the drug. Then the generic drugs come in, which are more cheaper. But this whole process, as we said, takes 10 to 12 years, and the patent life is 20 years. So you have only about 10 years. If you are so successful and so rapid and so fast in developing a drug, then you have about 5-6 years of getting your return on the investment that has gone into developing this particular new drug, and then all the failure rates that go with it. So this costs about, in today's terms, about $2.5 to $3 billion even, including the failures that go along with it. And I'll tell you how many do succeed and how many get failed on an average. So I will not talk about this slide. I did talk about the percentages and all, but this is purely without the failures. That is what it costs. If a drug straight away succeeds, it costs about $1 billion or more. But with the failure rates that come in about you lose about $1 to $1.5 billion of money when you are actually doing it. So actually, if you are looking at the drug discovery, if 10,000 compounds are developed, so in the preclinical stage, only 250 compounds reach. And when we are talking about clinical trials, only 5 go up there. So it's like a sieve, what you actually go through, and ultimately one drug gets approved by the FDA. So it takes about 10-12 years in getting this particular drug out to our patients. And that's how the drug development process takes a lot of time. Now, clinical trial process. Now, this was basically to give you an understanding of how the drug development works. Now, what is the clinical trial process? In the beginning, we talked about that clinical trials are the way it is designed is very important. But when we are running a clinical trial, if I have designed a protocol, I want to evaluate something, an existing treatment versus a new treatment or a new methodology, then what are the things that we should be very careful about? Now, it's not only people who are doing investigator-initiated research or consultants, but students who are getting for their thesis. A lot of times, as I've seen with a lot of our students, what happens is they just have to complete, they have a 3 to 4 year term and they have to complete all their, so it's in the last 6 months, 8 months, they try to finish it off because they have to get their degrees. But you know, a lot of effort actually goes, and if a little bit of thought process is given into it, a good publication can come out of every single study or the thesis that a student does. So reason for the way we are behaving today is because we are never taught like that. So I think it's important to really focus on this. And whether you do a hardcore clinical trial, whether it's an academic study, whether it's a regulated study, it really doesn't matter. The methods are the same. You're dealing with the same kind of patients. You're looking at answers that you are trying to see. So that's what research is all about. But so in that whole process, some of the things that are critical, that are important, which generally we are not aware or the students are not aware, I will address some of those points. So as you know, these are very general and everybody would have studied this in pharmacology, and probably this is the only thing that we read in our pharmacology, whether it's Goodman, Gilman or Oscar or whatever. We just talk about, oh, there are 4 phases of clinical trials, preclinical, then there is a phase 1, phase 2, phase 3. Phase 1 are safety trials where we try to find out, evaluate safety, gather information of how a drug interacts with the human body. Basically, you're looking at pharmacokinetics, pharmacodynamics of the study of a particular drug. Similarly, in phase 2, you're looking at safety. Safety is a constant phenomenon that you will look right through post-marketing phases, because safety of a drug is extremely important and critical in research. And then only you try to look at whether the drug is efficacious or not. So in phase 2, you increase the number of people, and then you look at monitoring of the side effects. You try to see whether what is the dose that would work best, whether this drug is effective. You try to look at, proof of principle, proof of concept. You try to do in phase 2. And then if you feel that it is effective and is safe at the same time, then you try to do a phase 3 study, which is basically a large scale study, which takes a few years. And then you try to, based on that result of that studies, which are called confirmatory studies or pivotal studies. If you succeed, you are able to make your endpoints and you are able to monitor safety at the same time. Based on that, you make an, registration application to the FDA, to the regulatory agencies, regulatory bodies for approval of that particular drug. So this is typically the typical phases of what clinical trials are all about. And in post-marketing, and a lot of these things, many of you might have heard, the post-marketing safety and efficacy studies have become more and more important. We call them real-world evidence generation, which can come from our day-to-day practice, what we see patients on a day-to-day basis. And we try to gather information on the drugs effect, because ultimately phase 1, 2, and 3 are very controlled clinical trials in a controlled setting. A certain number, there is an inclusion, exclusion. You will include some, you will not include, because you're doing by a strict protocol. And it has a different statistical sample size. So the exposure of patients is limited to maybe a few hundreds or maybe a couple of thousand patients. But when you are selling the drug, when you are giving the drug, prescribing the drug to so many patients, the sample size of your patient base is bigger, and they have comorbid conditions and other modalities. And that is the time you try to see what are the other effects of the drug. And you might have heard several times the drugs are withdrawn, they were found hepatotoxic, because those things didn't come up so well in our limited clinical research phases. So post-marketing is extremely important, and now it has become more and more important from an FDA, because everybody wants to try to reduce the costs, because these, as I mentioned, development of phase 1 to phase 3 trials is very expensive. So if you're able to gather a lot of information from day to day in our real practice, and then try to really generate evidence out of it, that information could go back in designing or even substituting phase 2 or phase 1 trials and actually go to phase 3 based on limited population of what you have known based on what you have gathered from the real world evidence. So it's becoming a very, very different domain, and that's where artificial intelligence and all is coming, because the huge amounts of data that we have across the world. In the real world scenario is huge and humongous. Now, why is human research highly regulated? We talk about regulations. When we're running a trial, we talk about ethics committee, we talk about whether it is regulatory available or not. Why is it regulated or why is it highly regulated? So past transgressions lead to need for laws that have protected. And we have had these experiences, and some of you might have heard about. There was a movie also on Nuremberg Code, the thalidomide tragedies which happened, which led to some of these amendments in the US FDA. It's all started there. The Tuskegee experiments on syphilis, which led to Belmont report, and some human radiation experiments, because these were done, gene transfer experiments. A lot of these experiments were done without any controls, without any ethics, without anything, any knowledge, and the rights of human subjects were not considered there. And that led to a lot of these disasters that we have seen. And that's why regulations came into place over a period of time. I'm not getting into the detail of that. Sometimes we might talk about what are the GCPs, why we have these, the good clinical practices, what are the ICH requirements. That we'll talk separately some other time. The informed consent, which is basically maintaining the rights of the patient. So whenever you are running a clinical trial, whether it is you are doing a study, which is, we typically say, hey, it's an observational study, I'm just recording the data, but everything is important. So you have to talk to the patient. Like when we are dealing with the patient in our routine practice, we do tell them that we are going to get you a PET scan and get them signed on all the possible good and bad things that can happen as a result of that exposure. So which is a consent that we take. Now here, informed consent from a trial or a study point of view is extremely important. And what should you be looking at it? Because a lot of times we do talk about, oh, is it consent required? Because I'm taking a consent anyway during my practice, because we are doing a procedure and they are taking, but this consent is very different. You're doing an experiment, whether it is observational or this, but you are using data of the patient, and with data laws and privacy laws, it's extremely, extremely important that these informed consent, as we call it, is extremely important. So what are those elements which are critical that we should be taking care of when we're looking at a consent? The research and the study purpose. Even if you are doing quality of life questionnaire, that is not a questionnaire or a survey. It is research where we are, based on which you are making decisions on health outcomes, based on which you are making certain decisions. That's why it's important. So research, study purpose, the risks, the benefits that you can have, alternative treatments. If you are giving a treatment or a therapy, what could be the alternates that you have to tell your patient that these are the alternative treatments? What are the confidentiality of records? Especially now when we are talking about genetics in cancer. So if you're doing whole exome sequencing, whole genome sequencing, now what are you going to tell them about what you find, along with what your purpose is basically, and are you going to disseminate those records? Because that can have a lot of important aspects are considered from an cultural, familial, social perspectives that you have to tell, and it can create problems for the patients. So a lot of times they don't want to go for genetic tests. Are you going to give that? So that's important, that we will be keeping your records confidential. Those will not be disseminated. Medical treatments available if injury occurs. Now, if you during your study, if something goes wrong with you as a part of this participation, what would you be given? Will you be treated for that? Suppose you have a serious adverse event, you have a problem, who will take care of that? So that should be a part of your consent. You have to mention what will be done if such thing happens, and whom to contact for answers to questions. You as an investigator should be available to answer those questions. So you have to mention who you should be going to. Statement that the participation is voluntary, that you are voluntarily participating. So you have right to withdraw. You can participate, you may not participate, but you are doing out of your own will. So these are some of the basic elements of an informed consent. Not that, okay, we are going through a procedure and you are okay, you may have X or Y. But these are some of the essential critical elements of a study informed consent document. And we can get into deeper details. It's a separate chapter in itself. But right now, these are the most important aspects that you have to be careful about when you are designing an informed consent in any clinical trial. The institutional review board or the ethics committees, as we talked, research ethics committees. Now, hospitals have medical ethics committees, but these are institutional review boards or ethics committees of the institution, research ethics committees. Now, you have to mention that all clinical trials must be approved and monitored by an ethics committee or a research ethics board. Now, a lot of times it's said that, well, there is hardly anything because I'm doing it in routine care. Ethics committee, institutional review board approval is essential and critical for every single study, academic, non-academic. Now, a lot of times there is a concept called exempt from review. Now, exempt from review doesn't mean that the investigator or the researcher is not going to go to the institutional review board and saying that, oh, I read that it is exempt from review. No, exempt from review doesn't mean that. It's the right of the institutional review board to decide whether it is exempt from review or not. So you have to make an application to ethics committee. And for all practical purposes, I have to tell you that now the whole process of integration between ethics committees or the institutional review boards, publication houses who publish articles where you will submit it for publication, and the clinical trial registries work together. So if you're submitting to a journal your study or you find some data which is of real value, you want to submit to them, the first question they will ask, please give us your ethics committee approval that you had in your institution or any IRB that you have gone to. Without that, we will not publish. Second, then they will also ask you about, have you registered your study in any clinical trial registry? So for important aspects, whether you do a retrospective study or you do a prospective study, it is important that before you start even a retrospective study or collate the data, you design your protocol, submit it to the ethics committee that you are going to do these 1, 2, 3, 4, 5. It is retrospective or prospective, and now get an approval for that. After that, then you can submit for publication. And when I say about publication, any indexed journal which is of good impact factor will not accept this without. And a lot of times students have come back to us saying that, oh, I submitted for journal publication, but they are asking for ethics committee approval. No ethics committee in the world or IRB in the world can give you a retrospective approval of any study, whether it is retrospective data or prospective data. So please remember this for your thesis. This is extremely, extremely important because we try to take this very lightly and we'll see, oh, my institution will give me an approval. No, institutional review boards are independent bodies. They are not institutional driven. There are external people sitting on those boards. So don't be under that impression. So this is an important aspect for research worldwide, anywhere. Now, when you have what we learned is that you have designed and you want to run a study, you have taken informed consent, now you have to get your patients into the study, whether it is academic. So most of the studies worldwide fail or not are completed because they are not able to recruit patients in a timely fashion. Only 3 to 5% of newly diagnosed adult cancer patients participate in clinical trials. This is hardcore data. And reasons for this relatively low number are many. We won't get into those because there are multiple aspects why a patient may not participate in a trial. So some of the recruitment strategies that you look at when you are running these academic or regulatory research studies, it's based on the physician who is deciding, or the investigator. The physician becomes investigator. That trust and the contact with the patient that they have, who are your patients who you are treating. The study staff who is a part of whole group. It goes from nurse to your research associates to your patient assistant to anyone and everybody to billing assistant, your whole study team or your whole team that deals with the patient. Speaking to community groups, sometimes you have to do community. If you're doing epidemiological research and try to find, or you're doing, research at, especially looking at risks of prevention and all kinds of those researches, which you have to go into the community. So it's important to build those community groups. Now, a lot of ads and radio ads are happening, internet websites, social media is being used, but there are certain limitations to it. Your ethics committee is to approve it, and then physician referrals. These are important because even if you're running within an institution, and patients go to different physicians, and you have to tell them that you are running a study for your academic reasons, and please refer patients. And these are, you give them details of that study. So getting patients in the study, then only your study will be complete. So this is again a part of your clinical trial. Now, when you do that, how do you get the data collected? Now, a lot of times we think, okay, we are running a study, I have designed everything, methods, materials, everything is there, but you have not really thought of what is the data that you want to collect, which is important for you. Because a lot of times we collect a lot of data and then don't know what to do about with that data. So you have to be very specific in your design of your study and your data collection, the pro forma as we call it in academic terms, but that's called the case report form, on which you report what aspects of your study, depending on what you are researching, will be collected. Much of the clinical data is taken from the subject's medical records, which is your source documents, which you have with you. And a lot of pharmaceutical and device trials data is verified by multiple players, that you do the source data verification, whether this patient is real and whether all this data does exist in the real files of the patient, the original files of the patient. So that subject data collection is very important. And then the analysis of the data, depending on what you have designed, you have taken statistical inputs for the design. So it's very important to look at it from that point of view as well. Now, while you are doing the studies, there might be adverse events, the side effects as we call them. I won't get into the deeper details, but these are critical ones, serious adverse events. These are not serious in terms of only severity wise, but they are serious because of regulatory reasons, and they are defined that results in death or are life threatening, results in hospitalization of a patient when you are doing an OPD kind of a study or recording data only, or if you're doing it in patients, then it gets into prolonged hospitalization, has a disability and has any congenital anomalies or birth defects in patients, especially in females who are of childbearing age. Events that are serious, unexpected, and related. These are some of the basic, these are regulatory definitions, but you have to be very cognizant during your study that I have to record that patient is doing well, patient is safe, and if there are any issues of their side effects, I need to note those down, and these are defined in your study. And then the clinical trial and product, as I said, depending on, you want to get up conclusions which are unambiguous. You're able to make very clear primary endpoints, which gives you the clinical outcomes of that test or the treatment or the device that you are looking into, or the method of surgical technique that you are looking at. So always strive for the ideal, but in most cases you have to settle for the best compromise, because every time it may not be idealistic. You have to look at what's the best way, and all of this has to be thought of right in the beginning. So when we run studies like this, we say that we have to design the study with an end in mind. So you work backwards, that I want this outcome. How do I come backward and actually design my study, design my protocol? What is my research question? And that's how you come to at an end. So always strive for the ideal. So the key message is, because you who is running the research is a primary clinical investigator, play a critical role in ensuring high quality study. Good care of patients is not the same as good clinical practices. Care of patients is good, but good clinical practice is how you record the data. You're following certain guidances. You follow good clinical practices. I would call it good clinical research practices, and ensure that all staff that is a part of your whole study have a clear understanding of the responsibilities under the FDA regulations. When I say FDA, we are talking about federal drug agencies of any country, which includes India as well. as a stake is public confidence, because ultimately what you come out with is applicable to patients. So public confidence and participation in clinical trials is ultimately the availability of safe and effective products. Okay, quickly I will run through study designs, some of these. We may not get into the deeper details because we don't have that much time, but I'll try to quickly go through some of the design. So when you have a research study design, it could be a descriptive, an analytical, or an experimental. Now, if it is a descriptive, we call it a case report, like what we report as case reports. We want to publish it. Case series. So typically we say up to 2 to 3 patients. If you have 2 to 3 case reports, then you call it a case series. Now, if you have 5, 6, 7, it becomes a small trial. It's not a series then. We sometimes it's a misnomer to say I have a series of 10 patients. It's a study in itself where the sample size is 10 patients. It's not a case series. And then you have cross-sectional studies, which are more descriptive. And then you have analytical, which we will talk about. Experimental, you have observational. And in observational, you have cross-sectional studies, case control studies, and cohort studies. Now, we may not get enough time to deal because this itself is a subject separate, topic in itself to get into different types of how we design and how we come to those. So, deciding the type of research study, did the investigator assign exposure? So, how do you design that? Basically, if you're trying to tell what is what. So, you are saying that if there is an experimental study, you do a random allocation. If there is a random allocation, you call this a randomized controlled trial. If there is no randomization, then you say a non-randomized controlled trial. And if there is no experiment, there is no intervention, then we call it an observational study. You have a comparison group. Then if there is a comparison group, we call it an analytical study. And that direction can be a cohort or a case control or cross-sectional study. And if it is no, then it is a descriptive study like what we said in the beginning, like a case report. So these are the different types of studies, but this is how we really assign those types of studies and how you decide what kind of a study it is. Descriptive study, describe the frequency, natural history, and determinants of a factor. This is just a definition of it. You identify patterns or trends in a situation, but not the cause and the effect of that. The case report, case series, and cross-sectional studies, which we mentioned. Case report is a descriptive study of a single individual. Case series, study of a small group, as I said, 2 to 3. And then cross-sectional studies and prevalence studies that examine prevalence, epidemiology, or survey of a disease or a clinical outcome. So those are kind of cross-sectional studies. Analytical studies used to test one or more specific hypotheses, whether an exposure is a risk factor or a disease or an intervention is effective in preventing or curing disease. So that's where you are trying to analyze and then examine the etiology and causal association of that study. Cross-sectional studies, it measures the cause and effect of at the same time, but does not tell us the relationship necessarily. And then it's conducted in a specific period which does not contain a follow-up. So you have a study group, which is whatever is your sample size. You talk about disease, you talk about healthy subjects, and onset of the study, you talk about end of the study. So it's just a cross-sectional study. You just follow it up straight away. Case control studies conducted retrospectively to inquire past histories. So typically cases already have a certain condition, and control would be without a condition, like healthy people we take generally. And the case and the control groups are compared for presence of certain factors. Like you have a case group, you have a control group, and you compare the two. And then you have a direction of the study. If it is either retrospectively, because you have enough of data which you are trying to see, I will look at a section of patients who were normal and a section of patients who will form as a control group, and a section of patients who have a particular disease that you are looking at or particular points that you are looking at. And if you are doing it prospectively, then onset of the study starts, and then you do it prospectively over a period of time. Cohort studies are follow-up studies, generally prospective and inquire about future. Begins with a group of subjects with some causative factor but free of the condition of interest. and investigate the effect of prognostic factors on a clinical outcome. So cohort group, you have factor plus and a factor negative, like a disease and a healthy, disease and a healthy, and then you do it in a prospective way. You start with an onset, and then over a period of time, you define the time frame depending on what you're looking at. Experimental studies are compare the effect of treatments or interventions with control in humans. It could be transparent, evidence-based, and randomized. Typically is used, which is supposed to be the gold standard. This is what it is, basically randomized control trials, which are supposed to be the gold standard in medical research. Patients are randomly assigned to a study group, and they produce the strongest evidence among the clinical trials. Two or more clinical treatments or interventions are compared. Okay, so you have a random assignment, which might be, patients are randomly assigned, you give a control group, then you have a follow-up, and then you compare the results at the end. So these are typically the randomized control. These are the gold standard based on which most of our evidence-based medicine and evidence-based treatments come into existence. The essential elements of a research protocol. Now, when you are designing a study, we set materials and methods, and what are those? What is that protocol? We typically call it in clinical research a standard protocol. So what is a protocol? The protocol demonstrates the guidelines for conducting the trial, the essential steps, and how the trial is to be carried out. It will define exactly what you are going to do in the study. It will talk about eligibility of the participants, the length of the study, the medications that you are using, the related tests that you will be doing in that particular study, depending on what you are looking at. It allows the researcher to plan and review the project steps, serves as a guide throughout the research, and forces time and budget estimates, because then you are looking at, okay, how long can I do the study? Will I have enough budget? Will I be able to recruit all the patients within that time period, depending on what types of patients you are looking at, what disease you are looking at. That's what your protocol exactly defines. Because typically we try to say materials and methods. It's okay, this is what I will do. How you will do, what all will be covered, is absolutely essential to be defined in the protocol. And then there has to be an objective and an aim to raise the question to be researched and clarify its importance. Why you are talking about this research? What will it lead to? So you have an hypothesis sometimes, and you want to say that, okay, this is my primary objective, this is what I want to achieve, this is my hypothesis, this is what I am trying to achieve. Collect existing knowledge and discuss the effort of other researchers. To formulate a hypothesis and objectives, to suggest the methodology, that's what we do, what we just talked about, and to clarify ethical considerations, what all. We talk about consent, we talk about approvals, we talk about what would be right, what would be a standard of care, what would be an intervention, all that you will define in that study. And to discuss the requirements and limitations and achieving the objectives. Sometimes, you may not be able to achieve, so you looked at the study design, you looked at the statistics, you look at those objectives, you look at the power of the study. All that has to be defined in that particular protocol, which we typically in our academic standards say, these are my materials and methods, but actually it's a protocol, and it should be well defined and defined to perfection. Protocol structure typically will have what I said is more defined introduction and a statement of purpose, the background of the study, why you want to do it, what is the clinical problem, what does the literature show so far, what has been done, what is your research question. Then you are talking about aims and objectives. What are you going to achieve based on what your clinical problem and justification is? What is the method? What is the design? In what setting you are going to do this? Sampling strategy, how you are doing, what are the inclusion and exclusion, what you will include, what you will exclude, depending on, like for example, we say patients have failed 2 therapies, 3 therapies, you're looking at immunotherapy, you're looking at new therapies. What would be that inclusion and exclusion? What will you include? Outcome assessments and measurements. What will you measure? How many times you will measure? Are you going to do PET scans? All these are important aspects that you will definitely define in your protocol. What is the data collection method? What statistical analysis and follow-up you will do? How you are going to analyze? What is those data points that you are going to capture and why? Then, methodological challenges like selection bias. How are you doing to random assignment, or are you going to randomize? Are you going to be doing it blinded or unblinded. Loss of follow-up. Patients are following up. You have given 2 cycles. You are expecting 6 cycles. What do you do? How do you define those? What would you consider in with patients might withdraw? Which is their right? Then feasibility of recruitment. Who will be the study team? Who is going to help you in the study team? Which are the participating centers? Are you collaborating with research? You do a lot of academic collaborative research also. And study funding and process. Who is going to fund my study? If you're doing additional things than what you routinely do with patients, how is that going to be funded? Are you going to do insurance coverages there or not? Because that's a requirement. Then study organization, ensuring data quality. Organization and management, how this whole thing will be organized. What are the responsibilities of investigator? Who will do the coordination? What are the ethical considerations? How do we ensure data quality? All the ethical consideration and the significance. So all of these are important aspects that you will mention. Typically, there is a standard protocol format, and it has different sections which have to be addressed. And some of the sections may not be applicable to a particular study, but that should be the basis, and not just a one-pager, study methods and materials and methods. That's how we put it in the journal. That's when you have done all of this, then you make an manuscript and then put it that way. So I will stop here. I think I have overspent by 5 minutes, because I think we will leave some time for questions. And though it has been fast, but I wanted to cover some of the basic items that are important, which you will come across, which we come across. And each and every one of these slides, I would say, will take one session in itself. So I'll stop. Thank you very much for your attention. So I'll be open for any questions now. Let me have a look at. I saw some of these questions coming up.
Thank you very much. It was a lovely talk, and it's one of the topic which students are never ever thought, never ever they concentrate and they suffer whole life. That is what unfortunately separates a country like India and other, where we have best clinician, highest clinical material, just because we are not good in data maintaining, auditing it, and we don't imbibe ourselves into clinical trial research publication. Nobody would take us seriously in spite of having the largest material. The big irony is we have the largest number of cervical cancer and the clinicians who do minimal access, and a trial is done by Westerners who do one surgery a year, and they write a guideline and we are asked to listen to them, which is biggest shame for a country like ours. Fantastic talk. That's correct. I think now you can open both Q&A and chat box and answer them, sir.
Okay, I'm just trying to get to the chat.
There is one question from Rakesh. Sir, how to define ethics in clinical trial? Is it okay just to write no ethical issues for an observation study, or any other detail way we have to mention?
No, that is a very I wouldn't recommend that. Now, no ethical issue is not the right thing to say. Like it is equivalent to saying that, like we say, no drug is safe. So we say, oh, the drug is safe, there is no side effect. So it's similar to that. So ethics, when there might be, see, normally when we are talking about a study, now you are undergoing a certain procedure, the patient is undergoing a certain procedure. Now, if you are doing an observational study, you will say there might be no additional risk over and above the risks that the patient is undergoing anyway because of the undergoing procedure. So that's how we would write. But the risk of any study would be even participation in a study. When you tell a patient that, it might give me, psychological pressure, and that itself being a part of the study. So what are you going to do me? That itself is a pressure in itself. So it's not that there is no ethical requirement, but you explain what it is. It might be minimal. You might say minimal risk, which is perfectly fine. Saying no is not a good option to say. So you might say that, okay, there is no additional risk. Yes.
When we submit a paper, they ask two things. One is no conflict of interest, no financial this one. Then they ask no ethical issue. So you would recommend that under the section of ethical issue, we would say that no further. What should we put it as instead of just writing no?
So in that, see, when you are writing in the protocol, when I told you there are different sections in a protocol, ethical section is one of the aspects which is required in a protocol. There we define what I just mentioned. Now, when you're submitting to a journal, So you will say there were no additional ethical concerns associated with this study, because there you will say, because conflict of interest and ethics and any funding thing is always one liner for the submission purpose for the journal, but in the protocol you will mention exactly what would be the thing that would
there is no trial which will not have an ethical issue. There will always be an ethical issue. So we can't say no ethical, but it was no additional extra ethical issue which were in this other than what is normal. Yeah, that's
correct.
Next question
as long as you have taken as long as you have taken the approvals and everything else, you're fine.
Okay. The next question is, what is the difference between a retrospective cohort study and case control studies?
Okay. So when you are doing a retrospective cohort study, now you are using a retrospective data, which is the data that is already existing. Okay. Now retrospective, there are two. We should not, we can define study because there are different types of studies, and one could be a prospective or a retrospective. That is different whether you are doing in future or you are doing past data. Now it could be cohort, it could be a control study. So every study would be like that only. So the definitions have to be done in such manner that okay, either a study is retrospective, like what we say, there is a study which is observational or interventional. So which means I'm doing an intervention, I'm doing an observation. Second is, am I doing a prospective or a retrospective study? So it could be a prospective observational, prospective or retrospective observational, or prospective intervention. Okay, because intervention can be prospective or intervention was from a retrospective. Similarly, then it could be a case control or it could be a cohort study. So there are different definitions which are, then you can club them together differently. So you can see what it is, because if you go back to the slides, if I go back to the slides, what is retrospective and what is prospective. Similarly, you have cohort, you have case control. So differences can be done in that fashion. So
case control study could be a prospective, retrospective. You can either do it broadly or you define a cohort of group, and then you can still do a case control study defining yourself a cohort. Then it could be retrospective or prospective.
Depending on what you are looking at, depending on what research question you have. Yeah. So the categorization of this is different. Yeah.
Okay. Next question is, as RCTs take long time, typically 5 to 7 years, sometimes 10 years for result to come, how effective are umbrella and basket trials?
Well, that is the new design that is coming. See, you can do a set of trials trying to get some of the, because when you are doing gold standard retro trials, that takes longer time. Now, depending on what your objective, as I said, even sometimes you are using a lot of real world data. So basket trials you are looking at different objectives that you are doing, or you are doing adaptive trials as well today. Now you might be adapting as you go, and you see it's taking time and you're not able to get the research. You try to design them, but all this, when you are designing this, you have to define this upfront. You can't do it retrospectively. All this has to be thought of, that okay, my whole design, my whole study will be in this fashion. So umbrella trials is a set of group of studies that you are trying to do and looking at different objectives that you are trying to achieve based on what you are looking at, what your objectives is, because you expect and anticipate I might get an answer, or it might take me longer. As I said, idealism cannot always work. So you try to design different designs. So basket trials are done in that fashion. Basically, objectives remain the same, but you have the flexibility of using and getting those outcomes which you could use and publish and talk about.
Then Ruchir Bhattacharya is asking, sir, can you please explain us level of evidence?
Level of evidence?
Yeah, level 1, level 2, level 2A, 2B, 3.
Yeah. There are different levels of this evidence. So I may not get into the deeper details of it, but evidence-based medicine has been based on different types of levels that are given to this, and it would all depend on the situation of what we are looking at, what kind of objectives we are looking at. But generally, it's a deeper discussion that we can have about it. I can explain that a little bit more if he can directly connect with me separately, then I can give her that discussion. I don't think right now we'll be able to finish everything.
So, Ruchi, if you see the level of evidence on NCCN is different. Level 1, 2A, 2B,
Correct.
2C and 3 they do. So level 1 and 2A are to be practiced in day-to-day practice. 2B is through an MDT. 2C is not outside a clinical trial. Third is an experimental. But there is another level of evidence. Then you just have 1 and 2 and 3. So by and large, phase 3 multicentric prospective RCTs go into level 1 if they are substantiated by meta-analysis. Level 2A is in the absence of a large multicentric phase 3 well-conducted randomized controlled trial, then only a meta-analysis of various studies or a large series of prospective non-randomized study at a multiple institute level which are replicated with the same result could sit very well into 2A. 2B are where there are two arms of treatment available and both are equally effective, but there is no comparative RCTs. 2C and 3 should not be practiced outside a clinical trial because they are not standard of care. Okay. Just read about it, and there are various sections as well. NCCN level of evidence is different, and in the DaVita, the clinical trial level of evidence of practice is different, but pretty much they all sit to the same. Okay, next question. Is ethical clearance required before a paper presentation in a conference?
Paper presentation in a conference. See, if it is a study that you have done anywhere in the conference, because study, ethical clearance is required for starting and initiating a study, whether it is a retrospective or a prospective study. So before presenting, you would have done the study and you have taken the ethical clearance. So that would have happened much before. So whether you present it as a poster, whether you present in any form, ethical clearance would be required right in the beginning itself. Now, if your project is exempt, still you would have got an exemption from the ethics committee that, okay, you don't need to take a consent, you don't need to be followed up and stuff like that.
So at the time of study, it is required, but to present in the conference, if you have never taken an approval from the scientific committee of the institution and the department, and ethical is ideally required before you collect the data to make a slide, ideally.
So every time you take an ethics committee approval for a study, now for presentation to a publication, to a poster or workshop or anything, you don't need an additional approval. You have taken an approval for the study conduct, data collection, based on which you are making conclusions. So that is only approved. So after that, nothing is required.
If it is not taken, please take it before you make the slide for the first time and you present it for the first time. Next question is from Megha Andrews. Sir, can you please tell types of randomization? Because I'm a beginner in the field of research, how it should be approached, like 1:1 randomization, double-blinded, and
Correct.
What is the types of randomization?
See, randomization is basically to remove bias in any study. So if you're sometimes you might have an open label study where you know what kind of a drug is there. Now, when you do the patient selection, it should not be that, okay, this patient I should give this drug, this patient I should give another drug, or I should do this procedure to this patient. So in order to remove that bias, you do the randomization because you are doing it by computer generated, so that, or saying that, okay, patient number 1 goes to A, patient number 2 goes to B, patient number C goes to A, like that. So randomization is to remove bias. Now, randomization can be blinded or unblinded. And at the same time, the randomization, if you are using a placebo in a drug, now in oncology, it may not necessarily happen because you don't give placebos in oncology unless there might be a standard of care and then you are adding another drug. Now, depending on the exposure and what your statistics tells you and the usage, because then you can do that, okay, my statistics tells me that I should give the active drug, the new drug, into a randomization ratio of 2:1 or 3:1, which means there are 3 patients who I will give active drug and 1 patient will be my control, which is my placebo. So that is a statistical requirement of any particular study that you will look at. And they said that, okay, this is my sample, I want to do it in this fashion. Or sometimes you will have, depending on what you are testing, 2 or 3 different arms you might have. So you will try to see that I have arm A, 1 dose, arm 2, B dose, and then I will keep 1 as a control. So it will depend on your statistics and your primary endpoints that you have designed in your study, that you will do a randomization based on 2:1, which means 3 patients will get ABC and 4 patients will be a control, like that. So it would depend on your design of the study and what your primary objectives and endpoints are in the study.
Thank you very much. A lot of questions keep coming, but we already crossed 8:30. As I always say, all good things has to come to an end, and the purpose of teachers, mentors, and the program is not spoon feeding our students, to stimulate their brain and show the direction. And now so many topics are excellently covered by Sridhar Care. Please expand on that, and we still have full year for you to complete the whole modules in the NVEMS. Don't worry. Keep attending the seminars, and there are dedicated course Dr. Sridhar Care does twice a year on what is a good clinical practice to conduct a clinical trial. And there's a beautiful 1 day or 2 day courses, certified courses he conducts. Please join one of them and learn it more extensively and expand based on what you have. Some people are interested to expand the statistics to be adopted in clinical trials. There is statistics courses we do through him. You can do it. Some people want to just get an overview. Some people want to look more at ethical, regulatory aspects. Please attend them. So this is just, you can't cover it for weeks together. These courses happen, and to cover in 1 hour is extremely difficult. Fantastic work, sir. Thank you very much. Heartfelt thanks on behalf of the board and me and the students. And thank you, and have a nice good night. I know it's late night there.
Thank you so much. Have a great day, everyone.
Thank you, sir. Thank you. And over to Navneet Singh ji.
Yeah, thank you very much, Professor Surinder here for the presentation, and thank you very much, Professor Somashekar, for joining us, and thank you, trainees, for joining the session.
Thank you. Bye-bye.